This is for adaptive immunity, specifically the second branch, the antibody-mediated immunity. Uh, so before we go into more details in the diagram, let's just get our orientation. This adaptive immunity has two branches, the cell-mediated and the antibody-mediated. The cell-mediated plays a role in basically getting rid of intracellular pathogens, so pathogens that make it into the cells. The antibody, which we are going over now, will be for extracellular pathogens that make it into the body fluids or in between cells, but not inside cells. So we can just think of antibody mediated as only for pathogens that are outside of cells. And these include also cells like macrophages, which I included because it's one of the starters. CD4 T cells, many may know these as T helper cells, and then also B cells. So just to get let's see what's going on in this diagram is that the phase 1, when I speak of this area, it's regarding this area here, phase 2, and then phase 3. So a little bit more to get more information, the antigen invokes an immune response, but what is it made from? It's made of proteins and large polysaccharides. Like, for example, on the microbial, there are cell walls, capsules, viral coats, flagella, and so forth that could act as an antigen to invoke an immune response. But in order to at the end of the antibody mediated, there's antibodies produced, but these are specific to only specific antigen receptors, which the antibody binds in this area called the epitome. So regarding the branch of antibody mediated, after phagocytosis, also know that the macrophage is the antigen presenting cell for phase 1 of antibody-mediated immunity. So the macrophage lays a piece of the pathogen, and it's called the foreign antigen, on its MHC type 1 and MHC type 2 markers. The MHC stands for major, H stands for histocompatibility, and C for complex. These MHC markers, number one, MHC type 1 are only located on all nucleated cells except for red blood cells. The MHC type 2 marker are located on professional antigen presenting cells like macrophages, for example, and others. Since the macrophages are or antigen presenting cells are also nucleated, they have both MHC type 2 markers, and they also have MHC type 1. But I left this really small. The reason is because in the branch of antibody mediated, the MHC type 1 marker does not play that much of a vital role. It has more of a role in when pathogens make it into infected cells. But since this pathway is regarding pathogens are outside the cells, it doesn't play that much of a role. But let's continue on. The macrophage presents the foreign antigen to a specific antigen receptor, and it's called the specific antigen receptor on the CD4 T cell. So this is the antigen, and this is the MHC type 2. You could think of this kind of like a Tetris. Let's say this closes, this antigen almost perfectly fits inside here, and this CD4 protein, which I will discuss in a second, almost fits perfectly inside here. Sort of like Tetris, just for diagram purposes. The third step is that the CD4 protein confirms the connection on the MHC type 2 marker here. The macrophage releases interleukin 1, and the fifth step is that the interleukin 1 signals the CD4 T cell to perform just two functions. It tells, which we'll discuss right now. The CD4 T cells basically those two functions is that it tells the CD4 T cell to release interleukin 2 and to make more interleukin 2 receptors, or actually make interleukin 2 receptors. So that's the fifth step. But the sixth step is that the CD4 T cells are stimulated when their own interleukin 2 binds to their own interleukin 2 receptors. Yeah. The seventh step is that the interleukin 2 signals the CD4 T cell to divide into two daughter subsets. So one is called the activated CD4 T cell, or is also known as Th2. The memory, and the other daughter cell will be the memory CD4 cell. It's also called Th2. Also, if you guys see yellow in this, or it's highlighted color, it means that it was actually made during antibody mediated immunity. So if it's highlighted, it's because it's made in this branch. If it's not highlighted, that means these are cells that were already in the body to begin with. Okay, so the eighth part is that after phagocytosis, so we're going to start the branch here again on phase 2. But before we get into phase 2, let's speak about these activated CD4 T cells. They're there for the initial infection. They only have a half-life they're actually very short. And they don't last very long, but the memory CD4 cells are there for years. So just in case this pathogen comes back, then the immune response will be able to basically react more faster. So now going on to phase 2, after phagocytosis, the B cell is an antigen presenting cell as well, but it's for phase 2 of antibody-mediated immunity. The B cell lays an antigen called T-dependent antigen. For now, let's say it lays it on its MHC type 2 marker. The T-dependent antigen, its purpose is that it depends on the activated CD4 T cell, so it can activate the B cell. Number 9 is that the B cell presents the T-dependent antigen to the antigen receptor of the activated CD4 T cell. The CD4 protein here confirms the connection of the MHC type 2. So again, think of this like Tetris, like this fits inside here, and then this fits inside here. So this looks like almost like a perfect box. The activated CD4 T cell releases interleukin 4 and interleukin 2, which signals the B cell to divide, ultimately to divide to a plasma cell and to a memory B cell. The plasma cells, this is regarding phase 2. Let's speak about the plasma cells a little bit more though. The plasma cells are larger than the memory B cells, but major thing about the plasma cells is that they have also specific antigen receptors, but similar to, you can see these receptors here. So they also have these specific antigen receptors, but the difference is that these plasma cells arise from B cells, yes, but these receptors actually, you could say, they float off, so they're no longer receptors, they're now specific antibodies. So now to review, this is for antibody-mediated immunity. For phase 1, the macrophage performs phagocytosis on a pathogen. So then you could say that so there's a series of steps actually in phagocytosis. It should make a different video for that one. So for now, the antigen, it is put on the MHC type 2 marker. Then the antigen is presented on to the specific antigen receptor. Then the CD4 protein confirms on the MHC type 2. So once this, you could say, Tetris box is completed, then the macrophage releases interleukin 1. And interleukin 1, basically what that does is that it tells CD4 T cells to do two things: to release interleukin 2 and to also express interleukin 2 receptors. And again, sounds odd, but the interleukin 2 binds to, that's released from the CD4 is T cell. It is binded to the interleukin 2 receptors. The interleukin 2 receptor, interleukin 2, basically tells the CD4 T cell to make two daughter subsets of CD4 cells. The first one is activated CD4 T cells, or Th2, and the other one is memory CD4 cells, for just continuous surveillance for later on. So starting over on the phase, but this is for phase 2, after the B cell, it's also a phagocyte, performs phagocytosis on a pathogen, let's say the same pathogen. It has a T-dependent antigen now. This T-dependent antigen depends on the activated CD4 T cells to activate the B cells. In order to do that, what occurs is that the T-dependent antigen binds to the specific antigen receptor. So you can see like a little box when that occurs. And then the CD4 protein communicates that the connection was successful, I guess you could say, and it does this on the MHC type 2. So now it looks like a perfect box. The activated CD4 T cells release interleukin 4 and interleukin 6 to ultimately tell the B cell to divide into two subsets of daughter cells. One is the plasma cell and one is a memory B cell. The plasma cell, again, has specific antigen receptors, but the difference between this type of receptors is that these specific antigen receptors release, basically detach themselves, and they're no longer receptors, they're now called antibodies.